Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. Now imagine that when the glucose level is down, right, or there is no supply of glucose. For example, the glucose, where the glucose will come? The glucose will come from the food, and as well as the glucose will enter into the cell, it will go through with these kind of reactions. But if there will be excess of fat, then these reactions will go in this direction, right, and these will go in this direction, and there will be less utilization of glucose. And if there will be less utilization of glucose, the enzymes which are being present within the glycolysis are going to be down regulated. Apart from the feedback mechanism or allosteric regulations, there is also another level of regulation is that you are going to have the regulation at the level of the protein synthesis, which means you are actually going to make the availability of these enzymes or the protein at lower level or the higher level, and depending upon the amount of these proteins or the enzyme, that particular type of activity is actually either will go up or will go down actually. So these kind of events are more relevant when you are talking about the bacterial system, because the bacterial system is going to have the polycistronic, the transcriptional unit, right, compared to that eukaryotic system has the monocistronic transcriptional unit. So when you have a polycistronic transcriptional unit, this means you are going to have the different types of enzymes being produced simultaneously from the single transcript. And in that case, all the protein synthesis of all these enzymes are actually going to be under tight regulation or tight control, so that you will be able to have the complete control over the different types of events such as glycolysis, Krebs cycle, and all other kinds of things, right. Although the Krebs cycle is not present in the bacterial system or the prokaryotic system, but for the sake of examples, there could be say many more other kinds of pathways. For example, you are going to have the fatty acid synthesis pathway, and you are going to have the other kinds of pathways like amino acid biosynthesis pathway and also on. So all these pathways require a very tight control, and one of the mechanisms through which the bacteria is actually bringing the control is by up regulating and down regulating the amount of protein what it actually going to synthesize. And this is all been achieved by putting these enzymes or putting these genes or putting the transcriptional unit under a complete control mechanisms, right. And all these are being a part of the operon, which means a system which actually is going to operate or going to regulate the transcription followed by the synthesis of these proteins. So the question comes, what is operon, right? So operon is a set of genes. So operon is a genetic regulatory system mostly seen in the prokaryotes and the bacteriophage, in which a group of structural genes are transcribed together under the control of a single promoter, which means the operon, technically the operons are mostly been present in the polycistronic transcriptional unit, and they are going to be present in the prokaryotes and the bacteriophage, where you are going to have the group of structural genes, or I will say the genes which is going to be transcribed for the different types of enzymes or the structural genes, and they will be under the control of a single promoter. This means this single promoter is actually going to have the control over the synthesis of these structural genes. So you're going to in a typical operon, you're going to have the promoter, right. Next to the promoter, you're going to have the operator, and the next to the operator, you're going to have this is going to be the coding region, right. So this is going to be the coding region, and this coding region is going to be responsible for. So this is the coding region, and then you're going to have the poly A site, right. So the coding region is going to be responsible for the synthesis of the A protein, B protein, and the C protein. So generally operons are very common in prokaryotes and the bacteriophage, but it is also found in some eukaryotes. The main difference is that the expression of prokaryotic operon leads to the polycistronic messenger RNA, whereas the eukaryotic operons leads to the monocistronic messenger RNAs. In this particular lecture, we are mostly being focused on to the operon, what is being present in the prokaryotic system. We have not discussed about the operons in the present in the eukaryotic system. So, the idea is that we should be able to tell you the concept of the transcriptional activations and transcriptional regulations, and how the things are being done. Similar kind of things are also being done in the eukaryotic system. So, prokaryotes are the single cell organisms lacking a true nucleus and the membrane bound organelles. It adopts the operant system as a mechanism to efficiently regulate the gene expression in response to the changing environment conditions. Environment condition means the requirements of the different types of metabolites, availability of glucose, availability of oxygen, and so on. So, the bacteria is, since bacteria is a single cell organisms, it actually gets affected very oftenly, and it is actually has to respond to these changes. Operon system is a genetic regulatory system found in the prokaryotic organism that allow the multiple gene with the related function to be controlled as a single unit. The system offers different advantage for prokaryotes, like it is energy efficient, right, because you are supposed to only synthesize the operator or the repressor, and that actually is good enough to control and regulate the transcriptional activity of several genes. So, operon system allows them to coordinate the expression of multiple genes involved in a common pathway and transcribe together as a single messenger RNA, saving the energy and resources by producing the necessary protein only when required. Then it's actually going to have a rapid response to the environmental changes. So, the operon system enable them to adapt to the changing conditions quickly. If condition change, the expression of the relevant gene can be turned on or off rapidly. It's simple and compact, right? So, prokaryotes can use a single regulatory region to control the expression of multiple genes. This is particularly advantageous in an organism with a small genome, where saving space is very crucial. So, if you require multiple genes as a regulatory genes and so on, you are actually going to increase the size of the genome, and that the bacterial system cannot afford, because bacterial system has to conserve the energy, conserve the space, and all those kind of things, and that's why it is important that it should actually operate and control the multiple genes with the help of the operons. Then it has the coordinated regulations. It allows the bacteria to go with the coordinated regulation, because it can regulate these 3 genes, 4 genes, 5 genes, which are actually being present in the single pathway. For example, if you are talking about the glycolysis, you are going to regulate the hexokinase, you are going to regulate the pyruvate kinase, and so on. So, since all these genes are present in a single operon, probably it is easy for the bacteria to manage all these, and on the other hand, it is also going to save the energy. So, there will be a coordinated regulation. So, this coordinated regulation ensure that the product of genes are produced in the appropriate stoichiometric ratio, which means if you are processing the single glucose molecule, you require 1 glucose molecule of hexokinase, you require the 1 glucose molecule of aldolase, and so on. So, you can actually be able to produce these proteins and enzymes in a right proportion, so that you should not waste the energy by producing one or other in an excess amount, and on the other hand, you should not have the lower production of any of these proteins. Then you have the resource allocations. When a particular nutrient is available, the genes required for the utilization are switched on. Once the nutrient becomes scarce, the operon can be turned off, preventing the wasteful production of the unnecessary proteins. Then we have the adaptation to the niche environment. So, the operon system enable the prokaryotes to adopt in a specialized niche by fine tuning the expression of genes that are specifically relevant to those conditions. So, this is also very important, that you are actually having a very fine and regulated balance, and that's how you can be able to have the fine tuning of the expression of the genes that are relevant for that particular environmental conditions. Then you have the evolutionary advantage. Organisms with ability to regulate gene expression rapidly and efficiently in response to the environmental changes were more likely to survive and reproduce. So, operon, as I said, is been proposed by the Jacob and Monod, and it has been control mechanisms through which the prokaryotic system is controlling the different types of gene expressions within the bacteria. So, in 1965, the Nobel Prize in Physiology and Medicine was awarded jointly to the Jacobs and Monod for discovery concerning the operon and the viral synthesis. So, this is the all scientists who have got the Nobel Prize in Medicine in the 1965 for their concept of the operon. So, before we get into the detail of the operons and how and we are going to take up some of the examples of the operon, it is important to understand what will be the general structure of an operon. So, this is just a general structure of the operon, right? You're going in the general structure, what you are going to have, right? So, this is the transcriptional unit, right? This is the transcriptional unit where you're going to have the promoters, you're going to have the operators, you're going to have the structural genes. For example, in this case, this is a structural gene for A, B and C, and apart from that, you're also going to have the regulatory genes. So, regulatory genes are actually going to be a part of is going to produce the regulatory proteins and is going to produce the regulatory proteins, and these regulatory proteins could be activators, right, or it could be repressor, and condition of the activator and repressor will go and bind to the operator, right, and so this region what you see here is actually a part of operon. So, regulatory gene is not going to be a part of operon, and regulatory proteins are going to either activate or either going to facilitate the binding of RNA polymerase, or it's actually going to have the other way around, right. So, regulatory gene encodes for a protein called regulatory protein, which either act as a repressor or activator which controls the operon, but it is not a part of operon because it has its own promoter, right. So, regulatory genes are not a part of operon. This is the part of operon where you are going to have the promoters, operators and the genes for the structural genes. Now, let us talk about the regulation of an operon. So, there are two types of transcriptional regulation which is going to be possible in the operon. One is called as, the first is called as the negative control, and the second is called as the positive control. So, then the negative control, in which the regulatory protein is act as a repressor which binding to the DNA and inhibiting the transcriptional of the protein, right. So, and then you have a positive control in which the regulatory protein is acting as an activator which stimulate for the transcription. So, this is the regulatory gene from which you are going to have the regulatory proteins, and regulatory proteins could either be a repressor, which means once it goes and bind to the operator, it will not allow the RNA polymerase to go and bind to the promoter, and that is how it is actually not going to allow the transcription of. So, there will be no transcription of the structural genes. This is going to be called as the negative control. Whereas, in the case of positive control, you are going to have the regulatory proteins which are actually go and bind to the operators, and that is how they are actually going to facilitate the efficient binding of the RNA polymerase to the promoters, and that is how it is actually going to have the more production of this particular structural genes, and these are called as the positive regulations. This means when you have this, you are going to have the more production. When you have this and you are going to have the lower production, this means it is going to be negative regulation, this is going to be a positive regulations. Within this, you are going to have the two different types of conditions, either it going to be inducible or it going to be repressible. So, operon can also be either inducible or repressible. So, inducible operons are those in which the transcription is normally been off, which is not going to take place, and it needs inducer to induce the transcription, which means it is going to be turned on. Repressible operons are those in which the transcription is normally been on, which means there will be a basal level of transcription which is going to take place. Sometimes it may happen to repress the transcription or turn it off. So, you are going to have the positive control, negative control, and then you can also have the inducible operon or the repressible operon. So, this is what I have summarized here. So, you are going to have the negative control, you are going to have positive control, and within the negative control or the positive control you can be having the inducible operon or the repressible operon. So, in the negative control, the product of the regulatory gene inhibits the transcription. In the positive control, the product of regulatory gene is going to activate the transcription. Whereas in the inducible operon, your initial condition, or I will say the basal level of transcription, is going to be off, which means you are not going to have the transcription of that particular gene. But once this inducer is present, then you are going to have the operon which is going to work. So, it is going to have the turn on the transcription. Whereas in the case of repressible initial condition, the basal level, there will be a transcription, but when the repressible is present, it is actually going to turn off the transcription. So, let us first discuss about the negative inducible operon. So, within the negative you can have the inducible, you can actually having the inducible, or you are going to have the repressible. Even within the positive control also you can have the inducible or the repressible. So, there are several different conditions in which all this has to be understood, right. So, let us first take the first example, that is the negative control inducible. So, in a negative inducible operon, in a negative inducible operon, the regulatory gene encodes a repressor which readily binds to the operator as operator site overlap with the promoter site, so that the binding of the repressor physically block the binding of RNA polymerase on the promoter and prevent the transcription. So, for the initiation of transcription, something is needed to prevent the binding of the repressor at the operation site and represent the operator's site of binding, that is the inducer. This type of system is said to be inducible, since transcription is usually off and must be turned on. So, in this negative inducible operon, what will happen is that you are going to, from the regulator, you are going to have the repressor, right. So, this is a repressor molecule which will go and fit and sit onto the operator. If the no inducer is present, this repressor will be keep binding to the operator molecule, and it will not allow the transcription of the structural gene, because repressor will bind to the operator and it will inhibit the transcription. So, there will be a transcriptional off, right. And when the inducer is present, what will happen is that the inducer is suppose the insulin, for example, right, or I will say glucose, right. If the glucose is present, what will happen is that the glucose will go and bind to this repressor, right, and in that case it is actually going to make the active repressor to inactive repressor, and then inactive repressor would not be able to bind the operator, and as a result it is actually going to allow the transcription of these particular structural genes. So, this is an example of, or the mechanism in which the negative inducible operon is going to operate. We are going to take the few examples and then you will be able to understand this more nicely. And then we have the second condition. Second condition is that you are going to have the negative repressible operon, right. So, the regulator gene in this type of operon synthesizes and the inactive operon that cannot bind to the operator. So, RNA polymerase readily bind to the promoter without any inhibition and transcribe the structural genes. To turn the transcription something must be needed to make the repressor active. A small molecule called a co-repressor binds to the repressor and make it capable of binding to the operator. So, in the absence of inducer, regulatory genes are producing the repressor, but these repressors are inactive, which means they will not be able to bind the operator, and that is why there will be a transcription. So, transcription is on, right. So, transcription is on, transcription is on, right. under the bacil level, because your RNA polymerase will go and bind to the promoter. There is no inhibition because the repressor what you are producing is inactive, and that's why there will be a production of on, right. So, there will be a transcription on. When the inducer is present, the inducer will go and bind to the repressor, and that's how it is going to convert the inactive repressor into a active repressor, and active repressor will go and bind to the promoter, and that's how it is actually going to inhibit the transcription, and that's how it is actually going to have the turn off. It's going to turn off the transcription. So, this is another example or another way in which the operon can be regulated. So, this is called as negative repressible operon. Then the third condition is the positive inducible response, right. Remember that in the positive it is going to be transcriptionally off, and then it is going to be on when the inducer is present. So, in a positive inducible operon, transcription is usually turned off because the regulatory proteins, that is the activator, is produced in an inactive form, right. Remember that when you are talking about the positive regulation, it is going to be inducer, it is going to be activator rather than repressor. So, the negative control, it is going to be done by the repressor, whereas here it is going to be activator. So, whatever we have discussed in the case of negative operons, it is going to be exactly the reverse. So, in this case the activator is produced, but that activator is in the inactive form, which means it cannot activate the transcription, right. So, transcription take place when an inducer has become attached to the regulatory protein, rendering to the regulatory site. So, when the inducer is not present, the regulatory region is producing an activator, but this is an inactive regulator, which means it requires some kind of modification so that it will go and bind to the operator, and that's how it actually can enhance the production or enhance the transcription. So, there will be a transcriptional off, right, because the this activator is not competent enough or efficient enough to induce the transcription. So, inactive activators cannot activate the transcription, and that's why there will be no transcription. But once you add the inducers, these inducer will go and bind to the activators, and once the activator binds to the inducers, they will actually going to have, there could be a structural changes within the activator, and that's how they will be actually go and bind to the operator, right, and the active operator stimulate the transcription, right, and that's how you are going to have the transcription of the structural genes. Then we have the positive repressible operon. This is exactly the opposite of the negative inducible operon. Okay. So, a positive operon can also be repressible. The regulatory protein is producing an activator, and that will bind to the DNA, meaning the transcription usually take place and has to be repressed. Transcription is inhibited when a substance becomes attached to the activator and render it unable to bind the DNA. So, transcription is no longer stimulated. So, this is exactly in the case of no inducer, you are going to have the active activators, and active activator will go and bind the operator, and that's why there will be enhanced production of these particular genes. But when the inducer is added, inducer will go and bind to the transcription, making them an inactive activator, and inactive activator will actually going to turn off the transcription. So, these are the 4 different conditions in which the operon can be regulated by the activator or the repressor proteins, and it could be inducible or it could be repressible. So, these are just the summary of what we have discussed so far. So, you going to have the repressible operon, or you are going to have the inducible operon. The repressible operon generally normally keep the synthesis on, but can be turned off by the repressors, whereas in the inducible operon generally the transcription is off, but can be turned on by the inducer. Repressible operon are mostly been present in the anabolism reactions or anabolic reactions, whereas the inducible operons are always been present in the catabolic reactions. In repressible operons, you are going to have the inactive form, whereas in the inducible operon you are going to have the active forms. and the examples of the repressible operon is the tryptophan operon, whereas inducible operon, which is the example of the lac operon. So, we are going to take up these examples so that it will be easy for you to understand what is mean by the inducible operon, what is mean by repressible operon, and so on. So, let us take a first example, that is the lac operon, and then we are going to take the tryptophan operon. So, this is the example where you are going to have the synthesis, whereas here you are going to have the breakdown of the substance. So, this is going to be related to the catabolic reactions, and this is actually going to be related to the anabolic reactions. So, let us first start with the lac operons. So, lac operon or the lactose operon, which is in short it is called as lac operon. So, the lac operon of the E. coli contains the gene which are involved into the lactose metabolism. It is expressed only when the lactose is present and the glucose is absent. This is very important. Okay, if you have a glucose, the lac operon will no longer be active. Lactose can be broken down by the E. coli, but it is not their preferred energy source. They would much instead use glucose if it is available. Lactose can be broken down more slowly and with less energy than glucose. However, if lactose is the only sugar present, E. coli will immediately use it as a fuel. The lac operon contains the three structural genes LAC-Z, LAC-Y and LAC-A. Okay, so these are the three genes LAC-Z, LAC-Y and LAC-A, and all these three genes have their own individual roles. So, LAC-Z is called as the beta galactosidase, LAC-Y is called as beta galactosidase permease, and LAC-A is actually been called as beta galactosidase transacetylase. These genes are transcribed as single messenger RNA under the control of the single promoter, that is this promoter. The lac operon is typically being present as a shut off, okay, or repressed in a normal conditions, but can be activated in the presence of the inducer, which is called as lactose or allolactose. Thus, the lac operon referred to be an inducible operon. So, allolactose is a structural analog of the lactose. Okay. So, these are the structural genes, regulatory genes, and the regulatory DNA sequences and the regulatory gene which are present in the lac operons. So, you are going to have the three different types of structural genes. LAC-Z, which codes for the enzyme beta galactosidase, the cleaves lactose into the glucose and galactose. This enzyme also converts the lactose into allolactose. Then we have the LAC-Y, which encodes for the beta galactosidase permease, which transport the lactose into the cell. So, basically the LAC-Y is, so this is, if this is the cell, the LAC-Y is actually going to bring the lactose into the cell. Okay, and then lactose is going to be get converted into glucose and galactose, right, by the gene product of LAC-Z, right. So, it is going to be called as beta galactosidase, and LAC-A codes for the enzyme which is called as beta galactosidase transacetylase. It is not essential for the lactose metabolism, but appears to be play a role in the detoxification of the compound by transferring and the acetyl group. Then we have the regulatory DNA sequences. Remember that in a transcriptional unit you have the promoters, you are going to have the coding region, and then you are going to have the 3' poly A tail, right. Apart from this, you are also going to have the, because we are talking about the operons, so in this case you are going to have the operators. So, you are going to have the promoters followed by operators followed by structural genes followed by the poly A tail, right. So, the regulatory DNA sequences, you are going to have the promoters. So, the promoter is the binding site for the RNA polymerase which initiated the transcription of the structural gene. Lac promoter is a weak promoter. So, remember that when we were talking about the transcription, we discussed about the weak and a strong promoters. So, there are compositions which makes the promoter as a weak promoter or the strong promoters, right, because the strong promoters allows the efficient efficient transcription of the DNA, right? And it allows the very efficiently the RNA polymerase to go and bind and complete the transcription, whereas in the case of weak promoters, the melting of the DNA or the other kinds of activities is very difficult, and that's how it is actually going to have the lower efficiency and lower production of the RNAs. Apart from that, you are going to have the operators. So the operator is a negative regulatory site bound with the lac repressor protein. The operator overlap with the promoters. Then we have the cap binding site. The cap binding site is a positive regulatory site that is bound by the catabolite activator protein, or the cap. When the cap is bound to this site, it promotes the transcription by helping the RNA polymerase bind to the promoter. Apart from that, you are also going to have the regulatory genes, which is called as LAC-I. So the regulatory gene LAC-I transcribed and produced the lac repressor protein and inhibited the lac repressor transcription. In order to accomplish this, it binds to the promoter, partially overlapping the operator. When bound, the lac repressor gets into the way of RNA polymerase and prevents the operon transcription. But when the lac repressor binds with the lactose, it becomes the repressed. So there are multiple conditions, right? So you're going to have the in the absence of, so just to make it comparable what we have discussed, so in absence of inducers, so remember that lac operon is lactose is going to be an inducer. So in absence of inducer, or in presence of inducer. So when the lactose is not available, which means the inducer is not available, the lac repressor strongly bind with the operator and stop the RNA polymerase from the initiation of transcription. However, the lac repressor loses its capability to bind DNA when the lactose is present. It leaves the operator and float away, making it possible for RNA polymerase to transcribe the gene. So if the lactose is not present, right, then what will happen is that the repressor will not be able to bind the operator. Okay, so if the lactose is not available, right, the repressor will go and bind to the operator, and since the repressor is binding to the operator, it will not allow the RNA polymerase to go further to start the transcription. So there will be no transcription of the structural gene of lac Z, Y and Z. And when the allolactose is absent, the repressor binds with the operator, so the transcription cannot initiated by the RNA polymerase without any preventions. Now when the lactose or the allolactose is available, so what will happen is these inducer will go and bind to the repressor, right? So when they will go and bind to the repressor, allolactose or the lactose, they will no longer be able to bind the operator, and as a result what will happen is that the RNA polymerase will move and it will actually going to do the synthesis of the structural genes, and that's how they are actually going to produce the beta lactosidase and other kinds of enzyme from these genes. So when the allolactose bind with the lac repressors, the repressor cannot bind an operator, so transcriptional initiation by RNA polymerase without any preventions. Apart from the lactose or the allolactose, some of the allolactose analogs can also be used for the lac promoters or the lac operons. One of the very popular lactose analog is the IPTG, or isopropyl beta D1 thiogalactosidase. It is actually a inducer for the protein production, and that we are anyway going to discuss when we are going to discuss about the molecular cloning. Then we have the phenyl beta D galactose, which is called as phenyl galactose, and then you also have thiomethyl galactose or the TMG. So all these are some of the lactose analog. So either the lactose, allolactose or these analogs can be able to modulate the activity of the repressor, and that's how they can actually be able to have the effect on the into the lactose operon. Then the lac promoter is a weak promoter. It does not bind the RNA polymerase more efficiently on its own. It won't be able to accomplish much more without the help of the catabolite activator protein. High transcriptional levels are facilitated by the caps binding to a stretch of DNA right before the lac operon, right? So this is the cap binding region and this is the cap proteins. and cap proteins are actually going to bind by the cyclic AMP. So, E. coli produces a cyclic AMP as a hunger signal in the low glucose conditions. By attaching to the cap, cyclic AMP modifies the structure of the cap, enabling it to bind the DNA and stimulate the transcription. Cap is inactive without cyclic AMP. Only when the glucose levels are low, the cyclic AMP levels are very high, does cap actually activate. So, in the condition of low glucose, when the condition is low glucose, you are going to have the large quantity of the ADP, and ADP is going to be get converted into AMP, and this AMP is actually going to be get converted into cyclic AMP, and the cyclic AMP will go and bind to the cap region, right. So, it is actually going to bind the cap proteins, and once they bind to the cap protein, they are actually going to block the activity. So, when the cap attached to the cap, cyclic AMP attached to the cap and activate it, allowing it to bind the DNA. Cap helps the RNA polymerase to bind to the promoter, resulting in a high level of transcription, right. So, in a state of starvation, you are going to have a very high amount of ADP, and that ADP is getting converted into AMP, and then the AMP is getting converted into cyclic AMP, and in the case of low glucose, this cyclic AMP will go and bind to the cap, and as a result it actually going to activate and allowing it to bind the DNA, and cap is actually going to help the promoter to bind to the promoter, and that's how they are actually going to have the high level of transcription of these genes. When there will be a high glucose, so in the case of high glucose, there will be no production of cyclic AMP, and that's how there will be no binding of cyclic AMP to the cap, and as a result the cap will not going to help the RNA polymerase to bind to the promoter, and that's why there will be a low level of transcription. So, there will be high amount of lactoperon transcription are only possible without glucose. This method ensures that the bacteria only activate the lactoperon and begin using the lactose after exhausting their primary energy source, that is the glucose. So, if the glucose is present, it is actually going to block or it is going to inhibit the lactoperon activity, simply because it is actually going to does not allow the production of cyclic AMP, and cyclic AMP is going to bind the cap region or the cap proteins, and that's how they are actually going to facilitate the binding of RNA polymerase to the promoter. So, if we summarize all these conditions, what is the conditions? You are going to have the glucose absent, lactose absent, right. So, there will be no transcription, right. So, there are going to be 4 conditions. In 4 conditions, you are going to have, in first condition, you are going to have the glucose absent, lactose absent. In those conditions, there will be no transcription of the lactoperon, or there will be no lactoperon activity, because the lactoperon inducer is also absent and the glucose is also absent. So, although there will be a production of cyclic AMP because the glucose is absent, but since the lactose is also absent, the repressor protein will actually going to repress the activity or repress the production from the RNA polymerase. Now, there will be a second condition. So, second condition would be that the glucose is absent, but the lactose is present, right. In that condition, there will be a high transcription, right, because in the absence of glucose, we discussed already, right, that from the ADP it is actually going to form the AMP, and from the AMP it is actually going to form the cyclic AMP, and the cyclic AMP will go into the cap, and it will go and bind to the cap region of the DNA, and that's how it is actually going to facilitate the promoter. And since the lactose is present, lactose will also going to bind the repressor, and that's how it is actually going to remove the repression, and as a result it is actually going to have the allow the RNA polymerase to go for the transcription, and that's why there will be a high transcription level. Now, the third condition is that you are going to have the glucose present and lactose absent, right. So, if the glucose is present, it is actually not going to allow the production of cyclic AMP, and there will be a low level of cyclic AMP, so that the cap proteins will not be able to bind to the cap region, and that's how the RNA polymerase will not be efficiently be able to bind to the promoter, and that's how. And on the other hand, since the lactose is absent, the repressor will actually going to bind, and it's going to allow the operator, right. So, it's going to allow the repressor to bind to the operator, and that's why there will be no transcription. In the fourth condition, both the molecules are present, which means the glucose is also present and lactose is also present. In that case, there will be a low level of transcription, because since the lactose is, so if the glucose is present, there will be no cyclic AMP, so it's not going to efficiently allow the binding of the cap proteins to the cap region of the DNA, and that's how there will be a very low level of transcriptional activity from the RNA polymerase. Because the lactose is present, it will actually going to bind the repressor, and that's how it is actually going to destroy the inhibition of the operator, and that's why it's going to allow the RNA polymerase to go. But this level of RNA transcription would be less compared to the transcription activity what we have just observed in the condition number 2. So, if we summarize all these activities, summary would be that the lac operon contains the gene, those are involved into the lactose metabolisms. Lac operon is a negative inducible operon. The genes are expressed only when the lactose is present and the glucose is absent. Remember, this is very important, because the glucose is the primary metabolite or primary metabolite preferred by the cell, whereas the lactose is a secondary metabolite and it is not being preferred by the cell. So, it will not going to utilize the lactose until the glucose is absent. And we have already discussed that when the glucose is absent, it is going to have the synthesis of the cyclic AMP, and the cyclic AMP will go and bind to the cap proteins, and that's how the cap protein is actually going to help into the binding of RNA polymerase to the promoter site, and that's how it is actually going to have the higher production of the protein synthesis. The operon is turned off in a normal conditions. The operon is turned on and turned off depending upon the glucose and lactose level, the catabolic activator protein and the lac repressor. The lac repressor blocks the transcription of the operon by binding with the operator. In the presence of lactose, it stops acting as a repressor. So, catabolite activator protein act as an enhancer, activate the transcription of the operon, but only when the glucose levels are low. So, these are the 4 conditions what we have already discussed. If we have the glucose, we have the no lactose, then there will be no transcription, right, because the glucose is present and it is going to inhibit the production of cyclic AMP. Then you can have the both glucose and lactose present, then it will be having the low level of transcription. Then third condition where the glucose is also absent and lactose is also absent, then there will be no transcription. And then the fourth condition is that the glucose is absent but the lactose is present. So, in that case there will be a production of cyclic AMP, and the cyclic AMP will allow the binding of the cap proteins to the cap region, and that's how it is actually going to facilitate the binding of the RNA polymerase to the promoter, and that's why there will be a high level of transcription. And because the lactose is also present, it will actually going to block the repressor and laci, right, and that's how it is actually going to have the high level of transcription. So, this is the summary of the lac operon. Now, let us move on to the next operon, and the next operon is the tryptophan operon. So, tryptophan operon is a part of the anabolic operon, compared to that the tryptophan operon is a catabolic operon. So, the tryptophan operon found in the E. coli, it is a group of genes that encodes enzyme for the synthesis. So, remember that the first operon what we have discussed is for the breakdown of the lactose. It is going to break down the lactose into glucose and galactose, and that's how it is going to be drive the energy from the lactose molecule, whereas here you are actually going to consume the energy. So, this is actually an anabolic pathway. It is a anabolic or negatively controlled operon. It always remain on in the normal conditions and off when the tryptophan level is high. So, this is exactly reverse what we have just discussed for the lactoperon. Tryptophan does not need to be synthesized by the E. coli bacteria when it is present in the environment. Hence, the transcription of a gene in the tryp operon is turned off on the other side. When the availability of tryptophan is low, the operon becomes on and the genes are transcribed by a synthetic enzyme for the tryptophan synthesis. Tryp repressor does not always attach with DNA. Instead, it binds and inhibits transcription only in the presence of tryptophan. Tryptophan binds to the repressor molecule and alter their structure, which switches an inactive repressor into an active state. Thus, the tryptophan act as a co-repressor, right? Remember that this is very important. Tryptophan act as a co-repressor because it enhances the repression activity of the repressor. So, it's become converts the inactive repressor into an active repressor. One unique feature of the tryptophan repertoire is the attenuation. Okay, so like regulation by the tryp repressor, attenuation is a mechanism for reducing the expression of a tryp operon when the level of tryptophans are high. However, rather than blocking the initiation of transcription, attenuation prevents the completion of the transcription. So, this is a very unique feature of the tryptophan operon. Now, the structure of the tryptophan operon. So, there are 5 structural genes tryp E, D, C, B and A that code for the enzyme involved in the conversion of the corosmic acid to the tryptophan. Remember that we have already discussed about the tryptophan biosynthesis when we were talking about the amino acid metabolisms. So, tryp E actually codes for the enzyme anthranil synthase 1, tryp D actually codes for the enzyme anthranil synthase 2, tryp C it encodes for the enzyme 5-phosphorylanthanylate isomerase and the indole 3 glycerol phosphate synthase. Then tryp B is encodes the enzyme tryptophan B subunits, and tryp A is actually going to encode for the enzyme tryptophan synthase A subunit. The controlling site in the tryptophan lies next to the tryp E and consists of a promoter, an overlapping operator and a leader region or the tryp L. So, in these are the regulatory region, right. So, this is the operon, right, this is the operon and this is the regulatory region. You are going to have the promoter, you are going to have the operators and you are going to have the leader region which is called as tryp L, and then you are going to have structural genes like tryp E, tryp D, tryp C, B and A, and they are actually going to give you a polycistronic messenger RNA. So, it is also contains a repressor regulatory gene called tryp R. When the tryptophan is present, the tryp R protein binds to the operator, blocking the transcription of tryptophan operon by inhibiting the RNA polymerase binding. So, this is the repressor regulatory gene tryp R. So, when it is going to be produced, it is going to bind the tryptophan and that is how it is actually going to make the active repressor, and that is how it is actually going to allow the binding of the repressor to the operator region, and that is how it is actually going to block the transcription of these genes, structural genes. Reactions catalyzed by the enzyme synthesis from the tryptophan operons. So, this is anyway we have discussed in detail the biosynthesis. Ultimately, from the indole you are going to have the synthesis of the tryptophan, where you are going to have the activity of anthranil synthase, anthranil transferase, PRA isomerase, IgP synthase and so on. So, all these genes are actually going to be a part of the tryptophan operons. This we are not going to discuss in detail because we have already discussed these things when we were talking about the tryptophan biosynthesis. Now, tryptophan operon regulations. So, in the absence of tryptophan, which means then when the bacteria actually require the synthesis of the tryptophan, so there will be low tryptophan into the environment. When there is a little tryptophan or absence of tryptophan in the cell, in this condition the tryp repressor is inactive because there is no tryptophan available to bind with the repressor and activate it by the conformational change. So, inactive repressor cannot bind to the DNA, that is the operator, or block the transcription, which allows the operon to be transcribed by the RNA polymerase. So, once there will be an absence of tryptophan, which means there is a low tryptophan present, then the repressor is not going to be active, because it has to bind the tryptophan molecule to become an active repressor, and that is how it will not be able to bind to the operator, and as a result the RNA polymerase will go and bind, and it is actually going to do the transcription, and it will actually going to produce the polycistronic messenger RNA. So, there will be high level of gene production or the transcripts from the operon. Now, in the presence of tryptophan, when there will be high tryptophan, the things are going to be reversed, because if the tryptophan is present, it will go and bind to the repressor proteins, and as a result it is actually going to form the active repressor, and if the active repressor is going to be present, it will go and bind to the operator, and that is how it will not allow the RNA polymerase to bind to the promoter and to complete the transcription, and that is how there will be a low transcription in the case of the if the tryptophan is present. Okay. Then we have the tryptophan operon transcriptional attenuation. So, it is possible to obtain more strict regulation in E. coli by repressing the transcription initiation alone, but translation mediated transcriptional attenuation offer the additional regulation. The attenuation site in the tryptophan operon is situated after the transcriptional start site. More transcription stop here when the tryptophan levels are high. When the tryptophan levels are low or sparse, does transcription continue to produce the functional protein. So, in this case you are actually going to have the attenuation site. So, this is actually going to be attenuation site what is present. So, in the high tryptophan level, it is actually going to be transcribed, and it is actually going to produce a messenger RNA, which is for this particular protein. So, when the tryptophan concentration is low, the entire operon including the LIGO sequence is transcribed into a messenger RNA. When the tryptophan concentration is high, only the 140 nucleotide, which is only the part of LIGO sequence that precede the attenuator, are transcribed into messenger RNA, and the structural genes are not being transcribed. So, it is actually when you have the low level of tryptophan, the transcription will start from here and it will go all the way up to the end. So, it is actually going to have the full length messenger RNA, where you are going to have the leader sequences and as well as the RNA sequences of the structural genes. Whereas, when you have a very high level of tryptophan, the transcription will start from here, but it will only going to end up here, and you are only going to have the leader sequence of 140 nucleotide, and that is how you are actually going to stop the transcription of these particular genes. And this is a very unique phenomena what is only happening in the tryptophan operon, which is called as transcriptional attenuation. So, what is the mechanism? So, the operation leader sequence has a 14 codon open reading frame, codes for a leader peptide of 14 amino acid, which with 2 tryptophan codons. The mechanism of translation mediated attenuation depend on the fact that the translation in bacteria is coupled with the transcription. So, ribosome begins translating the 5' end of the messenger RNA when it is still being synthesized. Thus, the translation rate can affect the structure of a growing RNA chain, which determine whether the transcription can continue or not. The function of the leader sequence is to fine tune the expression of tryptophan operon based on the availability of tryptophan inside the cell. The 2 tryptophan codes for the leader sequence lies within the region 1, and the translational stop codon lies between the region 1 and 2. The leader sequence contains the 4 regions, that is the region 1 to 4, and that can form the various base paired stem loops or the hairpin like the secondary structure. So, regions are like you have the region 1, region 2, region 3 and region 4, and region 3 is complementary to both the region 1 and region 4, and as a result what will happen is that it is actually going to form a hairpin like structure. So, it is actually behaving exactly the same as we have discussed about the intrinsic transcriptional stop site, right. So, it is actually going to form a loop kind of structure, and as a result it is actually going to stop the growth of the RNA polymerase. If the region 3 and 4 base pair with each other, they form a loop-like structure called attenuator and function as a transcriptional terminator. If pairing occurs between the region 3 and 2, then no such attenuation forms and the transcriptional continues. So, this is exactly the site, right, what we have just discussed, right. The ribosome binds to the tryptophan polycysteinic messenger RNA that is being translated when the tryptophan levels are high and start the leader sequence translation. The translation stop codon is present between the region 1 and 2 and the 2 tryptophan codes for the leader sequence are within the region 1. The ribosome follows the messenger RNA closely during translation and creates the leader peptide. This peptide, the moving ribosome complete the translation of the leader peptide and pause at the stop codon, blocking region 2. At this point, the ribosome prevents the region 2 from interacting with the sequence 3. So, sequence 3 base pair with the region 4 to form a 3-4 stem loop which serve as a transcriptional terminator, and as a result, tryptophan prevents the tryptophan operon from continuing to be transcribed. So, this is what exactly happen when you have the high level of transcription tryptophan. So, there will be a 2 tryptophan coding region what is being present, and they are actually going to allow the formation of a loop like structure, and this loop will actually going to stop the progression of the RNA polymerase, and as a result it is actually going to stop the RNA polymerase and stop the transcription. So, this is exactly what we have discussed. So, if the tryptophan is in short supply, then the ribosome will pause at the 2 tryptophan codon contained within the sequence 1. This leaves the sequence 2 free to the base pair with sequence 3 to form the 2-3 structure, also called as anti-terminator. So, the 3-4 structure cannot form and transcription continues to the end of the tryptophan operon. So, when the tryptophan levels are low, there will be a base pairing of 2 and 3, and these 2 and 3 are called as the anti-terminator because it will not be able to find a strong loop structure, and that is how the transcription will continue, and that is how it is actually going to have the synthesis of the messenger RNA for the tryptophan synthesis. Now, let us move on to the third operon, and the third operon is called as the arabinose operon or the ARA operon. So, the 5 carbon sugar L-arabinose must be broken down by a operon known as L-arabinose operon, also known as the ARA or the ARA-bad operon in the E. coli. The 3 structural genes ARA-B, ARA-A and ARA-D are found in the L-arabinose operon code for the 3 metabolic enzymes needed for the breakdown of L-arabinose. These genes generate the enzyme called arabinose or the ribonucleokinase, ARA-A which is called an isomerase, and the ARA-D which is called an epimerase, which catalyze the conversion of the L-arabinose into the D-xylose 5-phosphate and intermediate in the pentose phosphate pathway. So, L-arabinose is also a part of the catabolic pathway, right, and it is going to follow exactly the same mechanism what we have just discussed about the LAC operon. So, a single transcript and messenger RNA is produced from the transcription of all structural gene in the L-arabinose operon. The catabolite activator protein or the CAP cyclic AMP complex, which is produced by the regulatory gene ARA-C, regulate the expression of L-operon as a whole. The proteins that codes ARA-C control the expression of ARA-bad by acting as both activator when the arabinose is present and a repressor when the arabinose is absent. ARA-C is sensitive to the level of arabinose. At high ARA-C level, the ARA-C protein not only regulate the expression of ARA-bad but also control its own expression. So, these are the metabolic pathway of L-arabinose by the action of 3 enzymes. So, when you have the L-arabinose, it is going to act by the L-arabinose isomerase, and that is going to convert the L-arabinose into L-ribulose, and then L-ribulose is going to act by the L-ribulose kinase, and that is how it is going to form the L-arabinose 5-phosphate, and L-arabinose 5-phosphate is going to be isomerized by the L-arabinose epimerase, and that is how it is going to produce the D-xylose 5-phosphate. So, what is the structure of the arabinose operon? So you're going to have the regulatory genes like ERA C, you're going to have the promoter which is called as para C, and then you're going to have the different types of structural genes like ERA B, ERA A and ERA D. Apart from that, you're going to have some of the regulatory proteins and all that. So this is the region of the arabinose operon, where the ERA B is going to be produced by for a ribulose kinase, ERA A is going to form the isomerase, and then ERA D is going to form the epimerase. L-arabinose operon is consist of 3 structural gene and a regulatory region with the operator region called ERA O, ERA O1 and O2, and the initiation region that is called as ERA I1 and I2. So these are the region, right? The structural genes are ERA B, ERA A and ERA D. And there is also a cap binding site where the cyclic cap and cyclic AMP complex bind to and facilitate the catabolic repression and result in the positive regulation of ERA B when the cells lack the glucose. The regulatory gene ERA C is located upstream of the L-arabinose operon and encodes the arabinose responsive regulatory protein ERA C. Both ERA C and ERA B have a specific promoter where RNA polymerase bind and initiate the transcription. ERA BAD and ERA C are transcribed in opposite direction from the ERA BAD promoters and the ERA C promoter respectively. Now, arabinose operon regulations, in addition to being under the control of cap cyclic AMP activator, the arabinose system is also positively or negatively regulated by the binding of ERA C proteins. So ERA C perform as a homodimers and interact with the operator and initial range region or initiation range of the arabinose operon to control the transcription of ERA BAD. A DNA binding domain and a dimerization domain make up such ERA C monomers 2 domain. Okay, so this is the DNA binding domain. So this is the arabinose binding site, and this is the structure of ERA C monomer, and there will be a dimerization. So you are going to have the arabinose binding site, and you are going to have the DNA binding site. The binding of arabinose is carried out by the dimerization domain. Upon binding to arabinose, ERA C undergo an endogational shift and adopt 2 different conformations. The binding of the allosteric inducer arabinose is also only factor that affect the conformation. When the concentration of ERA C rises too high, ERA C can potentially adverse auto regulate its own expression by attacking dimer ERA C to the operator region, and ERA C production is inhibited. So now you have the negative regulation of the ERA BAD, right? So cell do not require the ERA BAD product to metabolize arabinose when it is not present, right? So in the absence of arabinose, you are going to have the negative regulation. So dimeric ERA C therefore function as a repressor. One monomer binds to the ERA B gene's operator, while the other monomer bind to the remote DNA half site called ERA A. DNA loop is created as a result. The ERA BAD promoter cannot be bound by the RNA polymerase while in this orientation. As a result, the structurally inhibited transcription is blocked. So this is what exactly going to happen. You are going to have the operators which are actually going to dimerize, and that's how you are going to have the binding of the operators onto the arabin2 and arabin2 onto the DNA, and that's how it is actually going to form a loop like structure. And in this loop like structure, the RNA polymerase will not be able to bind. Then you have the positive regulation of arabinose. So both in the presence of arabinose and the lack of glucose, the arabinose operator is activated for expression. So when you have the low glucose, you are going to have the ADP followed by AMP followed by production of cyclic AMP, and that's how you're going to have the cyclic AMP cap proteins complex formation, and that complex is going to bind the cap region of the DNA. And on the other hand, when the arabinose is present, arabinose will go and bind to the operators, and that's how it will not allow the interaction of the operators to form the loop like structure like this, and that's how there will be a transcription of the structural genes from the operons. So ERA-C and CAP cooperates and act as an activator when arabinose is present. So when glucose is absent, a high level of CAP protein cyclic AMP complex bind to the CAP region site. Binding of cyclic AMP is responsible for opening up the DNA loop between the ERA-1 and ERA-2 O2, increasing the binding affinity of ERA-C protein for ERA-I2 and thereby promoting the RNA polymerase to bind to the ERA-BAT promoter to switch on the expression of the ERA-BAT required for L-albinose metabolisms. So this is all what we have discussed in relation to the regulation of the transcriptional regulation to the operons. Now what we have discussed that the operons are being functional mostly into the prokaryotic structure, but the operons are also present into the eukaryotic structures. And in a typical operon, what you have is you have a promoter, then you followed by the operators and followed by the structural genes, and these operons can be positively being regulated or the negatively being regulated, or they can be inducible or the repressible. So in this context we have discussed about the two operons from the catabolic reactions and one operon from the anabolic reactions. So in a lac operon, it is a catabolic operon where you are going to have mostly the operon is going to be present as the negatively regulated operon. So there will be no production of the proteins and there will be no production of enzymes, but when there will be an absence of glucose and there will be a presence of lactose, then the bacteria will actually going to have the transcription of these genes, and because the repressor is going to be bind by the lactose, and that's how it is actually going to relieve the inhibition, and that's how there will be a transcription of the gene, and that gene is actually going to act on to the lactose molecule, and that's how the lactose is going to be get converted into glucose and galactose, and that's how that glucose will be utilized by the glycolysis to produce energy. Apart from that, we have also discussed about the tryptophan operon, and we have also discussed about the arabinose operon. So this is all about the discussion about the operons and how the operons are actually regulating the transcriptional activity within the prokaryotic system. So with this, I would like to conclude my lecture here. Thank you.